Hi, my name is Kevin Saunders, and I am Director of Research for the Duke Human Vaccine Institute. Together with collaborators at Duke and Harvard, we have been working to design components of a highly effective HIV vaccine. An HIV vaccine that is broadly and potently protective has been difficult to make because our immune systems do not readily make molecules called broadly neutralizing antibodies, which inhibit HIV from infecting cells. To induce broadly neutralizing antibodies, we have realized that we must start to learn the rules of how the immune system works. Understanding the basic rules would allow us to safely guide antibody development down the paths antibodies would not normally take. By studying how highly effective antibodies develop in HIV-infected individuals, we have learned some of these rules and have begun to develop a HIV vaccine using them. Our article in Science shows that some of the main roadblocks in the development of HIV broadly neutralizing antibodies can be overcome. To overcome the developmental roadblocks, we designed particles with many fragments of the HIV outer coat protein called trimer nanoparticles. These nanoparticles bind well to progressive stages of antibodies journeying down the path towards broad anti-HIV activity. Specifically, we immunized genetically altered mice that we know have the human antibody precursors of broadly neutralizing antibodies. And we were able to stimulate these precursors to develop into more potent anti-HIV antibodies. Our team solved the structures of the antibodies that were induced by vaccination and showed them to be similar to naturally produced neutralizing antibodies in humans. Lastly, we vaccinated monkeys and generated antibody responses indicative of the beginning of broad neutralizing antibody development. These results are important because they demonstrate the feasibility of this vaccine strategy for guiding the immune system to make effective antibody responses. We envision that this strategy may also be used to make vaccines against other pathogens or malignant cells. I am delighted to be part of this project that highlights the strength of team science and underscores how basic science discovery fuels translational research.